The overall goal of the techniques presented here is to label and culture embryonic tissues for measuring morphogenetic deformations during early development. For culture of the early chick embryo, begin by removing the embryo from the egg using a filter paper carrier method. Depending on the experimental application, label the extracted embryo with fluorescent lipophilic dyes delivered via magnetic iron particles or with polystyrene microspheres. Next, track the labels during culture using fluorescence microscopy or optical coherence tomography imaging. Resulting morphogenetic strain maps indicate regions of tissue elongation, shortening, and shear in both 2 and 3 dimensions.
Hi, my name is Ben Filas, and I'm a graduate student in the lab of Dr. Larry Tabor in the Department of Biomedical Engineering at Washington University in St. Louis. Before beginning these types of experiments, it is important to consider relevant tissue culture techniques in your model system, imaging modalities to which you might have access, and also the type of data that you desire from your experiments. For example, are you interested in analyzing tissue deformations in 2 or 3 dimensions? We address some of these questions here today by showing 2 complementary labeling techniques in the early chick embryo.
Hi, my name is Victor Varner, and I work in the laboratory of Dr. Larry Tabor at Washington University in St. Louis. In both of these techniques, we quickly and simultaneously label hundreds of cells in the early chick embryo. Tracking the motion of these labels then allows us to not only determine the tissues into which these cells eventually take up residence, but also to quantify the tissue-level deformations that mold the early embryo.
First, incubate the eggs in a longitudinal orientation to the desired stage as defined by Hamburger and